Sì, sì, ok, dove andare. Ma poi va via. Ok. Okay, good. So, good morning to everyone, people here in the room and also in remote. It's a pleasure to introduce to you Cara Davis. Professor Cara Davis is Assistant Professor of Pediatrics in the Division of Hematology Oncology at the Stanford University. Cara obtained her bachelor degree in Pennsylvania and the doctoral degree at Philadelphia, and then she completed the clinical training in pediatrics again in Philadelphia and then in Stanford in hemato-oncology, right? And during her fellowship, Cara worked in the laboratory of Gary Nolan, and there she started utilizing a single-cell high-dimensional analysis platform to study healthy human B-cell development and B-cell leukemia with a specific translational focus. And in fact, her research is focusing on using single-cell analysis to study the organization of tumor heterogeneity in blood cancers to determine cell population associated with clinical risk, such as relapse. But she's also a medical doctor. She's doing clinical activities, seeing patients with leukemia, and she is involved in cancer cellular therapy programs in Stanford. We are collaborating since long time with Cara, almost 15 years. We just counted the years. She's a friend. It's curious that to tell you that one of our previous PhD Demon student, Yolanda Sarno, who is here, was working with Giuseppe Gaipa that started this collaboration at Fondazione Tettamanti, and she did the postdoc there, and she's still with Cara as a research assistant, research scientist, senior. And then now we have also another PhD student, Manuel Quadri, working in my group, doing some training activities in her lab in Stanford. So it's a pleasure to introduce Cara with the title of the presentation, which is Predicting Patient Outcomes from Single Cells in Childhood Cancer. So from the title, you can see that Cara will demonstrate the value of single cell, not only in research, but also for patient stratification and the potential application of single cell for treating human cancer. So thanks a lot for coming, and thanks for your presentation.
Thank you. I hope it's okay. I'm going to remove my mask for this part. Thank you so much for the invitation to be here with you guys today. It's been great to be back in Italy after many years and such a strange few years. So happy to discuss our work. So as many of you know in this room that childhood leukemia was universally fatal 100 years ago. This is a report from 1933, which really outlines a few patients who presented to this physician and described the rapid progression of the disease for children who had overwhelming amounts of white blood cells and succumbed to their disease within a few months. But we also now know that B-cell acute lymphoblastic leukemia, which is the most common type of cancer in children, is really an incredible success story in cancer treatment in general. These Kaplan-Meier curves are from the US studies, basically, which have shown both on the left-hand side here from St. Jude and on the right-hand side from the cooperative groups in the US, the incredible progress that is also seen in Europe as well with curing kids who have ALL. But we also, when you're in the clinic and you're treating these children, we know that relapse is still a major challenge. And in the United States, actually, relapsed ALL is the second leading cause of death from cancer. And up until a few years ago, it was still the first leading cause of death. So it's still a major problem, even though we do a very good job for the majority of kids. And these Kaplan-Meier curves just illustrate studies that were done sort of earlier in the late 90s, early 2000s, and the lack of progress. And then even more recently, this paper published in 2018 showing survival of patients after relapse. We haven't really made a lot of improvements. But many of you also may know, oops, sorry, I went too fast, about chimeric antigen T cells, which are really a revolution in the way that we treat many diseases, and there's a lot of promise in many diseases. And in ALL, which is one of the, really the one of the initial demonstrations of their promise, there's pretty incredible responses for patients who otherwise had no other therapies. But even in these curves, we can see that the event-free survival is only about 50%. And even with the updated version of this data, which was from the first multi-center trial of Kymriah, which is the Novartis CAR T cell, the 3-year survival looks about the same. So about 50% of patients are also going to relapse after getting CAR T cells. So the focus of my lab and our work in the lab is to really use single cell tools to uncover treatment resistance in childhood cancer. And so we think about this at the time of diagnosis. You have billions and billions of cancer cells. Patients undergo treatment, and you may or may not be able to query remaining cells or sort of early resistant cells. And then patients will go on to either be cured or potentially relapse from their disease. And we want to use single cell tools to look at this diagnostic sample or resistant samples so that we can, as early as possible, make a treatment decision about who may need something different. And the workhorse of our work has been mass cytometry, or CyTOF, which is really a single cell technology now that's been around for quite a while. But it's essentially flow cytometry that is coupled with mass spec so that you can measure dozens of proteins on individual cells, pretty easily, and compared to single cell genomics, a lot more economically. And so the way that mass cytometry works is, and the way that we use it, is that we often will stimulate cells in vitro, cross-link proteins with a fixation step, and then permeabilize cells so that we can actually expose intracellular isotopes, and we can ask questions about intracellular proteins. These are then stained with isotope-tagged antibodies and run through the instrument, which analyzes the cell by time of flight. So basically, you get data that looks very much like flow cytometry data, and you're actually measuring the time it takes for an isotopic mass to get to the detector. And so one of the very early challenges that we recognized in looking at cancer cells with single cell analysis is the extreme heterogeneity that you would get from looking at hundreds of thousands of cells from one patient, and then trying to expand that to many, many patients becomes quite challenging. And Yolanda and I just came from a single cell Keystone meeting in Florence, and it was quite interesting now that single cell technologies are really exploding. I think that this still is a challenge, and a lot of groups are still using single cell approaches like single cell sequencing to look at immune cells or healthy development in which the variability is much less, as opposed to actually looking at tumoral cells, which are still extremely heterogeneous. So one of the first questions that we had to answer as we were starting to generate data was, how do you organize such a very heterogeneous group into something that makes any sense? And so I like this analogy. I think about this a lot like a pile of Lego blocks, which you start out, looks like a big heterogeneous pile of Legos, or, also can be called a big mess. And the goal of taking those blocks is to build them into an interpretable model that you can actually understand and ask questions about. And so the thing that we rely on in building Legos, or also in, analysis with these single cell technologies, oftentimes is prior knowledge. So what has been published in your system? What do we know about, these blocks, for instance, and how should we be able to put them together? And so what is lacking often in cancer is what is the prior knowledge, especially on the single cell level. Whereas in healthy tissue systems or immune systems, we have a lot of information about how healthy cells develop. So this was our very, very early first attempts at mass cytometry with patient samples. These are 9 patients from St. Jude, actually, who all had Icaros mutations. And we were very interested in starting to look at that. And I will tell you a story about how that comes full circle in science life. But the first thing that we realized when we were just clustering these cells based on their expression of surface proteins, that basically every circle here represents a cluster of cells, but they're colored by patient. And what we could see was that every patient pretty much clustered together. There was very little overlap between the patients. And our goal was really to understand what are shared cell phenotypes that may have something to do with patient outcomes. So this, we encountered this challenge pretty early. I think we've come a long way, both in our lab and in the field. This is a little preview of a paper that's getting ready to be submitted, where we really have gotten to a place where we have a philosophy about how to approach single cell data and how to break it down in a way based on what are the questions that you're asking. So when you are generating single cell data, you can actually sort of look at biological phenomenon on 3 levels. One is on the actual single cell level. So what is happening in individual cells? The second is cluster level data or population level data. So maybe you're looking at and you want to say, how are the monocytes compared to the neutrophils, compared to the, whatever, megakaryocytes, let's say. And then the next level up is the patient level data. So how are you making these comparisons then across patients or cohorts? And all of these different levels of analysis have their own tools that are already available, their statistical challenges and sort of approaches that we think are best practices as you start to approach that data. So going back to our specific challenge in ALL, one of the ways that we approached this was taking what we know about normal development of healthy developing B cells, which is the cells that become cancerous in B cell ALL, and applying that as our scaffold upon which to model our leukemia data. So this heat map here is from a paper I published when I was in Gary's lab, where we used mass cytometry to look at healthy B cell development in human bone marrow. And we were really able to outline the developmental stages by surface markers, intracellular markers, and then also signaling and actually immunoglobulin rearrangement to put cells in sort of their developmental trajectory. So we took that scaffold of normal development and we published a developmental classifier now 4 years ago, hard to believe how fast things go, where the way that this worked was we took our healthy control bone marrows that we had also run with mass cytometry. We gated them along what we knew was the developmental trajectory of B cells. And then for each one of these populations, we assigned it an 11 protein barcode, essentially. We said, we want to know what's the expression of these 11 proteins in each one of these populations. And then we used that to take every leukemia cell, and we did this in 60 samples. Most of them were from here, actually. And we matched every leukemia cell to its most similar healthy population. And that was about 600,000 cells per patient. So we had a lot of data from every single patient. And this was published, like I mentioned, so you can look at this paper, but just briefly to set the stage for some of our future work, we used machine learning as a way to extract features from all of that single cell data to try to predict which patients were going to relapse in the future, even though we were taking this information from the sample at the time that they first walked into the hospital and had their bone marrow taken. And so that model, which we developed and called the developmentally dependent predictor of relapse, or deeper, basically identified 6 signaling features in cells that were transitioning between late pro-B cells and early pre-B cells, and that the relapse associated cells, or the cases, the patients that were going to relapse, had activation of these signaling molecules in these populations of cells in their diagnostic sample. And we were able to validate this on some paired diagnostic and relapse samples. And then when we looked at how does our predictor perform compared to standard clinical risk predictors, like the things that we as physicians use when a patient does come into the hospital, we found that actually our model deeper further stratified patients and actually added in a statistically significant way to the ability to determine who may be at higher risk of relapse and who is at lower risk of relapse. So we were very excited about this result. And to zero in a little bit on those cells that we found, this is sort of a fancy way of looking at the relationship between these signaling networks, which is conditional dependency analysis. But basically, to summarize what these pretty heat maps mean is that between diagnosis and relapse, what we recognized is that as these proteins were acting together in a network, so SYK, S6, and CREB, which are both SYK being proximal to the B cell receptor machinery, and S6 and CREB being very further downstream, either as transcription factors or readout of protein translation, that these were acting together in a network, meaning as one of them changed, the other two changed. and that their relationship to each other strengthened from diagnosis to relapse. So potentially suggesting that the cells, what I hypothesize is that actually at relapse, there's more homogeneity among the cells because all the cells that were susceptible to the chemo are dead, and it's only the resistant ones that are coming back, and they rely on a particular phenotype in order to persist. And so this is just what this data looks like on the single cell level as you would look at it on a flow cytometry plot. This is 2 patient samples from diagnosis on the left and relapse on the right. And we can see either, persistence at the relapse time point or even enrichment of these, triple positive cells at the time the patients relapse. So this left us at the end of this project with a model in which we hypothesized that this signaling network was calling out active signaling downstream of the pre-B cell receptor, which is a really important developmental checkpoint for B cells as they are normally developing. And so if you remember your immunology, B cells go through successive stages in which they have to make DNA double-strand breaks, they rearrange their heavy chain, and then they go through this moment where they sort of try on the heavy chain and see if it signals properly with the pre-B cell receptor. If it does, if it works fine, then they get to actually progress through development and rearrange their light chain and have the chance to be a grown-up good B cell. If it doesn't work, they get to try again. So this is a very important sort of fate decision that happens in the trajectory of a B cell. And so we were hypothesizing then that these sort of hidden cells that were there at the time of diagnosis, they have this activated network, and that what we're reading out is sort of constitutive activation of this signaling pathway. And we did see co-expression of the components of the pre-B cell receptor, and of immunoglobulin heavy chain in these cells as well. And that potentially then as patients undergo treatment, as I mentioned, all of these non-relapse predictive cells sort of go away. Perhaps MRD cells, or the cells that persist during the early weeks of treatment, are actually the same cells that we identified in the diagnostic sample. That's a question that we're investigating. And then that these cells can populate a relapse. So I already mentioned this because I forgot the slide was coming. But as I mentioned, again, this is a very important developmental checkpoint. And the other thing to note is that normally there are sort of these periods of differentiation in which the cells are not proliferating, and then proliferative periods where the cells are proliferating because they've sort of passed their checkpoint. So it's like you did it, you made your immunoglobulin, so now you can make more of your productive immunoglobulin, so you have more chances to have a good full immunoglobulin when they rearrange the light chain. And of course, this happens not in a vacuum. It happens in the marrow niche and with a lot of crosstalk with stromal cell populations and cytokines that are really important for these developmental processes. But we know that there's this, in this developmental moment, this normally very tightly regulated balance between IL-7 signaling and pre-BCR signaling. And there are these sort of crosstalk of these two signaling networks in the normal situation. So pre-BCR signaling has previously been implicated in ALL and also implicated in the risk of relapse or treatment resistance. Marcus Mushin's group, now over 5 years ago, published a paper where they used sort of genomic classification and then some mouse modeling to identify cases of ALL based on the ability of the cells to have this pre-BCR activation, and they called these pre-BCR positive and pre-BCR negative ALLs. And there are some enrichments of some particular genetic backgrounds in the pre-BCR positive, like the 119 translocated ALLs that have rearrangement of TCF3 and PBX1. So we then, I like to think about this as, in our case, our relapse predictive cells are cells that have this active, constitutive active pre-BCR signaling. And I think about this as the lights are always turned on and we can't turn them off. Whereas the remission predictive cells, or sort of the gray cells, are the cells that the lights are off, but you can turn them on because we've been able to show that those cells, we can actually activate their signaling cascade, but then it doesn't stay on. So in order for us then to really start to think about why do these cells actually persist and how are they treatment resistant? We went back and took 6 samples from our original cohort, sorted out their pre-B cells, and performed RNA sequencing. And we did this from 3 patients who were in the group that would go on to relapse and 3 patients in the group that would not. And we did find, of course, differential expression of a bunch of genes. And what was really interesting is that the pathways that started to come out in the relapse predictive cells as enriched pointed to a lot of metabolism and cellular metabolic processes that were focused around oxidative phosphorylation, reactive oxygen species, glycolysis, and fatty acid metabolism. And then, of course, we also did see some of the pathways that we saw when we were reading out the signaling pathways, such as mTOR signaling and PI3 kinase AKT signaling, which was expected based on our protein and the CyTOF data. So Dora Jidui is a really fantastic undergrad in the lab in collaboration with Yuxuan Liu, who's leading this project, went to the Broad cancer dependency map data set, where basically that has all these different cancer cell lines where they've done extensive CRISPR screens to look for dependencies. And so Dora went and she pulled out all the ALL cell lines, separated them into BCR positive, pre-BCR positive, and pre-BCR negative, and looked at their particular dependencies. And there were only 69 genes actually that were, had particular dependencies in the pre-BCR positive group. But when she put this into a Reactome database, again, these were identifying things that had to do with energy and OXPHOS and fatty acid metabolism, with a lot of mitochondrial gene lists and TCA cycle coming up. And so Yuxuan went to make some tools for us to then be able to interrogate this a little bit further. So hypothesizing that this is all happening with this activation in the pre-BCR, we went to the most upstream of our identified proteins, which is Syk. And she performed a series of, she made a series of Syk knockdowns in ALL cell lines in order to essentially be able to see what's the difference when you have this constitutive activation versus when you can turn that off with the knockdown. And so here's 3 examples of pre-BCR positive lines, 697s, NOM6, and Kasumi2, showing total Syk after the knockdown. And when she performed RNA sequencing on these cells now from the knockdown versus the wild type lines, and in conjunction with what we had from the patient data, again, we had the same pathways popping up in the RNA sequencing data, OXPHOS, mTOR signaling, fatty acid metabolism, as well as actually MYC and targets as being activated in the knockdown cells. Sorry, in the wild type cells, which corresponds with what we saw in the relapsing causing cells. So now we started to get into some very more basic biochemistry and cellular metabolism assays. And I don't, for time, because I want to tell a few stories, I'm not going to get into all of it. But to summarize, what we're starting to see is that the cells that are pre-BCR positive and are sort of dependent on that signaling actually have different energetic demands and flexibilities. So I had to go back and watch, 4 hours of Khan Academy videos so I could remember cellular metabolism. But basically, remembering that there are many ways cells can make energy. There are many substrates with which they can use, both through glycolysis, which does not go through the mitochondria, but then also many different substrates that go into oxidative energy production in the mitochondria, including fatty acids, as well as proteins and amino acids. And so what we found, to summarize here, in the dependency of pre-BCR positive versus pre-BCR negative lines, when we did some assays in the seahorse, was that the pre-BCR positive cells preferentially prefer glucose, then fatty acids, but they actually have, the ability much more easily switch in what substrate they're using compared to the pre-BCR negative lines. So they seem to be able to be more flexible in their metabolic use compared to the pre-BCR negative lines. So potentially one reason why they may be more resistant is if you, deplete or impact one, they just easily switch and use another. So Yuxuan looked at this also in a platform that we're developing in conjunction with Sean Bendel's lab and also Ravi Majeti's lab at Stanford to actually now look at this on the single cell level. Because when you do these assays on the seahorse, which is one of the sort of main assays people use for metabolism, you need millions and millions of cells. And that's hard to do, especially when we know that the cells that we're looking for are not the majority of cells necessarily in a cancer sample. So she went to some expanded samples from PDX. So these are primary cells that were expanded in mice. And she categorized them based on their pre-BCR status on flow cytometry. So pre-BCR negative lines or patients, 4 here, and pre-BCR positive patients, 5 here, where we can see they actually express IgM and 179B, which is part of the surrogate light chain, and looked at their expression. And I'm sorry, this is like a Mac to PC thing, but because the labels overlap. But basically, the cells, especially from the patients, if she gated them based on sick as our readout of their pre-BCR activation, that there was a relationship where the activation of downstream pre-BCR proteins in the purple is also coordinated with the activation of oxidative and mitochondrial proteins like ATP5A, which is a mitochondrial enzyme, and IDH2, which is also a mitochondrial enzyme. So that, the activation of the signaling couples with oxidative metabolism. And so to kind of summarize this part, one of the things that she's working on now is an approach using CyTOF to be able to look at sort of all of these things at once in primary patient cells. So our signaling molecules that we know are important for relapse prediction, our developmental molecules that we also know are, we need to focus in on particular developmental states, and then a whole slew of metabolic markers and readouts so that we can basically take what you would normally do on the seahorse and do it in single cells in primary patient samples. And so we have in the lab right now about 130 samples that are waiting for this. That's a combination of samples from the Children's Oncology Group as well as St. Jude. And we have clinical outcomes on those patient samples so that we can start to put all of this together, in addition to doing some more of the work with the actual knockdown cells and doing some traditional metabolomics with mass spec on those cells as well. So I'm going to transition to talk about another story, which this particularly is Yolanda's work that I want to highlight. And so again, thinking about treatment resistance, we all know, or many of us know, that glucocorticoids are really a mainstay of therapy for ALL. And I think for me, I forget that glucocorticoids, they actually play a job in cells, and really one of them is to regulate gene expression. And glucocorticoids, in the way that it works, is that the receptor is intracellular. A glucocorticoid goes into the cytoplasm, it attaches to its receptor, that translocates to the nucleus, and then it can exert its effect on gene expression in a couple different ways. One can be direct binding onto DNA of the bound receptor. The other can be binding to DNA in conjunction with transcription factors, or it can be binding to a transcription factor and then the transcription factor binding to the DNA. And these glucocorticoids and the GCR together can either be activating of gene expression, or it can be repressive. So it's very heterogeneous and context dependent. And we know through the history of clinical trials in ALL that patients with the ALL who get 1 week of steroids, they can actually, that response to 1 week of steroids can determine their risk of relapse. And this is very well studied, and actually through the trials that are run here in Europe, as almost I think about it as a early form of minimal residual disease that was happened upon. I think for time I'm going to skip through this, but just to say that there's also been a lot of work about metabolism and the relationship between cellular metabolism and energy, and how that regulates both the expression of the glucocorticoid receptor and also interplays with B cell transcription factors that have to do with sort of energy metabolism. So all these things, they start to become connected in some way. So as I mentioned, this is Yolanda's work, and it started a very long time ago, just too long ago, where we had, after looking at our progression of development in healthy B cells, we were really interested in understanding what are the transcriptional programs that sort of change across these developmental stages. And so we performed RNA-seq on sorted cells from these 3 populations and in 3 individual healthy donors. And in doing pathway analysis of these data, what we found was that as expected, as the cells developed, so from top to bottom, that we saw activation of B cell receptor signaling, PI3 kinase signaling, IL-7 signaling pathways, B cell developmental pathways. But one of the pathways that popped out was glucocorticoid receptor signaling, which was not expected and not really understood if that has anything to do with B cell development. There's been like 1 or 2 papers in mice, and mostly in like peripheral B cells, not early B cells. So Yolanda took 3 more healthy bone marrow donors, developed a glucocorticoid receptor antibody and readout for the CyTOF, integrated it into our ALL panels. And indeed, we found that the glucocorticoid receptor is expressed on the protein level. It's expressed most highly in pro-B cells and in coordination with sort of early B cell markers. And it kind of starts to go down by the time the cells are moving past into the pre-B cell stage. We know that glucocorticoids regulate cell cycle. So in these healthy progenitors, Yolanda looked and found that it was the cycling cells in S phase and in G2 that expressed the highest levels of the GCR. And that glucocorticoid treatment, in this case, we always were using dexamethasone, induced cell cycle arrest. So once these cells saw glucocorticoids for 48 hours, they were more likely to arrest in G0 and G1. So we wanted to then look at this in ALL cells, knowing, of course, the importance of glucocorticoids in ALL. And so we started out with 2 lines that we had, 2 cell lines that we had in the lab, NOM6 and REH cells. The NOM6 cells, after 48 hours of dexamethasone, about 40% of them died. And the REH cells were pretty much tolerant. They didn't die, they didn't do much. And similarly, the NOM6 cells, we saw the same cell cycle arrest. So we went to take a look. The REH cells actually, in the native REH cells, they actually have a mutation in the glucocorticoid receptor so that they don't express it. And so here's the protein for REH compared to NOM6. So Yolanda overexpressed it. She took our REH cells and overexpressed the GCR, and that enabled those cells now to become sensitized to glucocorticoids. So now, about half of them died. After 48 hours, we saw the same arrest in cell cycle after we reintroduced the receptor. And what we found, though, is that it's actually the pro-B cells that were preferentially killed compared to the pre-B cells in both cell lines after glucocorticoid treatment. So again, integrating some gene expression analysis, we took those surviving cells from these cell lines and did RNA sequencing. And we found that, after glucocorticoid treatment, cell cycle pathways were downregulated, not unexpectedly from what we know about glucocorticoids, but the pathways that were enriched in these resistant cells had to do with B cell development and B cell receptor signaling. Again, sort of implicating this developmental signaling in potential resistance. Yolanda then just decided to see if we could target that activated signaling by adding dasatinib, which will, again, target a very upstream protein of the pre-BCR, which is SRC, and added dasatinib into culture with these cells. And we could see that actually there was, with increasing dexamethasone and dasatinib concentrations, we had decreased viability in both cell lines with the addition of dasatinib. So suggesting that with the resistant cells adding this inhibition of that signaling, we could get improved cell killing. So we went to the freezer and pulled out some primary samples to see how does this look in primary patient samples. And so we had 19 primary samples, 14 from diagnosis, 5 from relapse, and 2 sets were pairs from diagnosis and relapse, and always a healthy control. These cells were treated with these drug combinations ex vivo and then analyzed on the CyTOF. And again, in primary ALL cells, we saw the same developmental patterns of GCR expression, such that the glucocorticoid receptor was most highly expressed in the pro-B cells and with the sort of earlier B cell markers. But we found, of course, and not surprisingly, that the level of expression on the pro-B cells was very heterogeneous. And of course, across all populations, but in particular, interest in the pro-B population, where it was the most highly expressed, but on a per patient basis, it was quite different. We did notice, though, that the highest levels of expression in the pro-B cells came from patients who were either a known poor prednisone responder from clinical, determination, or patients that had sort of bad genetics. So when we started to look now in the patient samples, what were the hallmarks of the surviving cells? We could see that initially, in all the patient samples, dexamethasone in general causes a decrease in viability. Dasatinib does, but not to the same extent. And the addition of dasatinib also killed cells, but it wasn't really amazing after 48 hours in these primary samples. But what we did notice was that, this, again, not every patient had cell death. And in fact, there were some patients who pretty much had, the same levels after dexamethasone treatment of surviving cells. And so we divided the cohort into responders versus non-responders based on that degree of cell death after 48 hours of dexamethasone. And we found that the GCR level was actually higher in non-responders compared to responders, and that it was actually that the responders tended to start out with less pro-B cells than the non-responders. So potentially something about sort of the developmental state that you're starting with and the level of glucocorticoid receptor in playing a role in the response. We put these into our classifier and basically found that both in responders and non-responders that dexamethasone, preferentially killed these late pro-B cells, but the cells that survived actually had a phenotype that was more like a later stage of development, like pre-B cells in both cases. And when we looked at the differential expression between the cells from these groups, the surviving cells from the non-responders really seem to upregulate proteins that are actually supposed to be repressed by the GCR. So things like NF-kappa B, cyclin B, and MYC. So suggesting that there's something sort of broken, potentially, internally in these non-responder cells in which they express really high levels of the GCR as if the cell is trying to do what it's supposed to do with the response, but it can't actually execute the downstream programs. We wanted to then ask the question, does this happen in patients? Because patients who are treated on the AIEOP treatment protocols used to get 1 week of steroids. And we had samples from 9 patients at time of diagnosis and then 8 days later. And we could see actually that indeed at day 8, we saw the same sort of contraction in this earlier population, but an enrichment in cells in this more mature population in the pre-B2 population. And that some of the phenotypic and signaling hallmarks that we expected did become different at day 8, such that cells had, higher levels of CD19, lower levels of CD10, and activation of some of the downstream markers like S6 and ERK on the day 8 sample. So Yolanda wanted to ask, because this is not something we do clinically, if we add dasatinib in a preclinical model, so in a mouse model, if we could actually improve survival and decrease engraftment. And indeed, that's what happened. So using a combination of dexamethasone and dasatinib, we found that in mice, and this is in a NOM6 model, that here looking at bioluminescence data of engraftment in the first 2 weeks, that the combination group with dexamethasone and dasatinib, the mice had decreased engraftment. And we can see this on the organ basis here in the bone marrow, as well as in the spleen. And that with this combination therapy that we are able to abolish the signaling that we came to know to be indicative of pre-B cell activation. We saw the same sort of developmental change where these cells seem to shift into a more mature state. And it did give a survival advantage in the in vivo setting as well. So to end this part, what we really found was that the glucocorticoid pathway seems to be coordinated with a B cell development pathway in normal cells. And this seems to be important in regulating the pre-B cell checkpoint. In leukemic B cells, you also see, we know that there's this expansion at this pro-B to pre-B transition, and these cells maintain developmental expression of the glucocorticoid receptor. But the cells that actually survive after glucocorticoid treatment seem to be pushed to a more mature state in which they may be more reliant on the pre-BCR signal, and that we can actually improve or abolish that reliance by treating them with a targeted therapy like dasatinib that will turn off that signaling. And this can actually result in a survival advantage in a preclinical model. Okay, last story. So I talked at the beginning about CAR T cells as being a really exciting new therapy in ALL, and also though the challenges in that, CAR T cells, even though they are fantastic for many patients, that we still see relapses. And one of the most common types of relapses that we are observing is when patients relapse with ALL that turns off CD19. So essentially, the T cell goes in, it's looking for CD19 positive cells. The ALL escapes by turning off CD19. And there's been a lot now published about why this happens. There's been reports of frame shift mutations in CD19, transdifferentiation into the myeloid lineage. So cells start out lymphoid, and after CAR T cell therapy, they become myeloid. You can have disruption of 19 getting to the surface, and as well intronic and splicing events in CD19 that make it no longer be susceptible to the CAR. So we wanted to ask the question, in this context, could we identify tumor intrinsic factors that are going to determine patients' risk for CD19? And of course, CD19 relapse, sorry. And this is relevant not only for CAR T cells targeting CD19, but there's also drugs targeting CD19 like blinatumomab that are used in the clinic as well. And this work is all led by Pablo Domizzi, who's an instructor in the lab. So in collaboration with Steve Grupp and Dave Barrett at Children's Hospital of Philadelphia, we had access to over 40 patient samples who were treated with CD19 CAR T cell and who were in different clinical subgroups based on their outcome. So patients who had complete remission after CAR, patients who had CD19 negative relapse, patients who had CD19 positive relapse, and then some patients who didn't respond at all. And of course, we used the CyTOF as our sort of normal approach. Pablo first shot an analysis, went and looked at the developmental classification of these cells. And before he started with the patient samples, he had CRISPR'd out CD19 on some cell lines and basically showed that between a wild type and a knockout CD19 cell line, there was no real change in developmental classification. But that was really not what we saw on the patient samples. So in our initial cohort of 8 patients, 4 pairs, we of course saw the enrichment of cells in this pro-B moment. Oops. But what we saw was this enrichment of cells in the post-CD19 sample where they had already relapsed, where there was this enrichment in early non-B cells, which is sort of a non-lineage population that we defined, based on what our panel was. And so the classifier relies on CD19 for its classification or its labeling of cells. And so Pablo took CD19, in silico out of their classification and reran the classification. And what we found was this population that we call pro-B2 discordant cells, in which if you keep CD19 in their definition, they're called pro-B2 cells. If you remove CD19 from their definition, they're early non-B cells. And so this started to explain why we were seeing this enrichment in these early non-B cells. And so we went back to the pre-CAR sample for all of the patients to see how frequent were these pro-B2 discordant cells in every patient. And they actually were in every clinical group, patients who were in CR, patients who had 19 negative relapse, 19 positive relapse. And they also were in a frequency between, 5 and about 20% de novo ALL, so patients who never saw a CAR first at diagnosis. But we didn't see them in healthy bone marrow. So we knew that these cells were present, but what was different about them in patients who were going to have a 19 loss relapse? And so based on CyTOF, when we started to look now at the single cell features of these cells, we found that the patients at risk for 19 negative relapse, their cells had lower levels of transcription factor Ikaros and PAX5. They also had lower levels of NF-kappa B as well. So Pablo went and used AbSeq or CyTOF on a subset of these patient samples, in particular, patients who were going to go into complete remission or those who were going to have a 19 negative relapse, and found on the RNA level that in pro-B-like cells from the single cell transcriptome, they also had lower levels of Ikaros on the gene level. A word about Ikaros, I mentioned it a little bit earlier. It's a transcription factor that's really important for lymphopoiesis, and it plays a role in chromatin remodeling and transcriptional regulation, and is really important in lymphoid specification. And it's previously been implicated in relapse risk in ALL through mutations or splice alterations. And so it's one of the things that I think is probably looked at in the clinical lab here in order to make risk determinations for patients. So we asked the question, does lower Ikaros translate to lower CD19? So Pablo took 2 approaches in 3 ALL cell lines where he either did shRNA knockdown or proteolysis with lenalidomide, because lenalidomide targets Ikaros for ubiquitination. And by both methods, we could see that after Ikaros knockdown, either through drug modulation or shRNA silencing, that as Ikaros goes down, CD19 also goes down in both cases and in all 3 cell lines. So Pablo took his cell lines and went on to do a multi-omics approach, doing RNA-seq and attack sequencing to look at chromatin accessibility in these cells. And what we found was that Ikaros doesn't affect CD19 mRNA levels, which is here on the left, or it does not affect the availability of the CD19 promoter. But it did seem to change splicing regulation in the cell. And so looking at differential expression analysis now between the wild type and knockdown cells, we could see that in the that Ikaros positively regulates in the comparison a lot of pathways related to mRNA processing and the spliceosome and mRNA splicing, which is not a role that Ikaros is sort of known for at this time. And indeed, when Pablo went back and started to look at splice isoforms and variants in his knockdown cells compared to wild type, we could see that there was actually a higher frequency of CD19 retention of intron 10, which would be a mis-splicing isoform, and would result in the premature expression of a stop codon in CD19. So potentially suggesting that 19 is could be being turned over faster, which is something he's working on asking experimentally. But also, if we look more globally at the RNA-seq and the attack-seq data, we also found that the Ikaros knockdown cells, as not unexpectedly, but they seem to have more genes that are associated with myeloid lineage. So as we know, Ikaros is really important for sort of enforcing lymphoid identity. When Ikaros is lower, these cells look more myeloid, or they start to express more genes that are related to a myeloid identity. So that could also, start to explain the risk of that myeloid transformation, which sometimes happens after CAR. So what we asked in collaboration with Maria Caterina Roti and in Robbie Maisner's lab was, did our Ikaros knockdown cells respond as well to CAR T cells? And so this data is in vitro. So we put our knockdown cell lines with CD19 targeted CAR T cells. This is all the mock CAR, but on the left-hand side of both graphs is with the actual CAR T cells. And compared to wild type in blue, the knockdown cells in red had the CAR itself had lower levels of IL-2 production and interferon gamma production, suggesting it wasn't being stimulated as well by the knockdown cells. And when we looked at killing capacity in an incucyte assay over 72 hours where the cells are co-incubated, we could see that the wild type cells are pretty efficiently killed with the normal CD19 CAR, whereas the knockdown cells, the CD19 CAR can't quite get around those cells and killing those cells as efficiently. So suggesting that the model is such that for patients going into CAR T cells, there may be these cells, which we call ProB2 discordant cells, that express CD19 at that time, but they have a propensity potentially to drop CD19 based on the level of Ikaros that they have in the cells, and maybe also in collaboration with other transcription factors like PAX5. More to learn. But this has to do with splicing alterations and loss of B cell identity, putting patients at risk for CD19 negative relapse. So I hope that I've been able to show you that using normal cellular development is a great way to organize really messy single cell data in cancer. And I think what's been really exciting for me sort of scientifically is in the last few years and how these different stories are coming together, specifically just in ALL, is that we're starting to see these pathways that play together developmentally across these developmental stages with the relationship between signaling, regulation of gene expression, possibly by splicing and regulation of different splicing, cellular metabolism, and chromatin state, all as being important components biologically that are important in normal development, but of course can be corrupted in the cancerous state, and really is an opportunity for us to learn about both things, and that we sort of are touching on a lot of these different quadrants in the 3 stories that I've told you today. What's next in a couple minutes? So we're really trying to develop new tools to improve the analysis of mass cytometry data. I mentioned this framework that is in preparation to be published on the right-hand side. On the left is a paper that just came out earlier this year, where we're working on batch correction and being able to use public mass cytometry data sets, which has previously not been possible. We are also really interested in, of course, analysis of more ALL samples and types and integration of mutational data, because that's a really important part that is sort of we haven't touched upon yet. So how can we do that? And using sort of more multi-omics approaches. Expressing antigen loss in the context of other immunotherapies. We have a project also starting out looking at CD22 loss after CD22 CAR. And then, of course, tackling other cancers, including AML and solid tumors in kids. So 2 more things, and then I'm going to finish. We've been working with Sean Bendel's lab on trying to identify surface markers that are unique to our relapse predictive cells by using a really novel CyTOF surface screen that looks at, basically it's the BioLegend screen adapted for the CyTOF, so 350 surface proteins. So we've done this now on some primary samples in PDXs, and indeed we can find surface markers that are unique both to relapse predictive cells as well as to sort of remission predictive cells in this context. And this is work being led by Dora Jadwi and Abhishek Kobalida. And then Yolanda's next trick, hopefully, will be looking at MRD cells, which is a project that she's been, had for a long time, collected samples when she was here and putting them on the CyTOF. And we have some really, I think, exciting early data and approaches, working with a really talented computational student in the lab using neural networks for the predictive modeling in these cells. And we already have some preliminary data showing that neural network modeling can really start to call out relapse predictive cells very early in these both diagnostic and MRD samples. And then last data slide, I mentioned that we're doing some solid tumor work. So we are using a similar approach to the CyTOF, but this is on solid or tissue sections called multiplexed ion beam imaging. It's like immunohistochemistry, except the antibodies are also tagged with a metal and read by mass spec. So this is like hot off the machine. We have a collaboration with John Maris at Children's of Philadelphia. We have over 100 neuroblastoma samples on a tissue microarray. And so we're looking at both immune infiltration, surface marker expression, but also the composition of neuroblastomas on the single cell level for adrenergic versus mesenchymal cell fate, and how does that relate to outcome for kids with neuroblastoma. So this is 4 different patient samples, and you get these very pretty images. So with that, I want to say thank you. Thank you so much for the opportunity to be here today, and here's some pictures of the lab having fun when we could during COVID. And of course to collaborators and funders, and to all of you for being here, and of course for the friendship and the collaboration now over 15 years, which I can't believe it's been that long. So thanks so much. And any questions if we have time? Okay.
Any question here?
Oh, yeah.
I can ask you a question.
Please.
You showed the data on Icaros. You know that Icaros, at least in the IOBFM protocol, is used as a stratification marker, not only ICROS itself, but it's a combination of different deletions.
Yeah.
You have a chance to look whether ICROS itself or the combination of gene deletion could be
important.
important.
Yeah. So we're looking. So not only Pablo's been trying to make some constructs in order to actually mimic the common gene deletions, but also we do know from the patient samples that we've looked at, actually what's interesting is that the patients who are refractory to the CAR seem to be enriched for Icarus mutations that we know of. Not every patient in our cohort did we know their mutational status. And then Pablo, actually, we just spent a month downloading the target data. So we're going back to the target data set to look at Icaros in the context of CD19 and CD22, actually. Yeah.
Tara, probably I did not get exactly what was the conclusion, if any, on the group of patient with CD19 positive relapse. Did you identify any signaling profile or signature?
Right. So I didn't talk about the CD19 positive relapse patients because we actually had very few in that cohort. I think we had 4 pre-diagnosis and 2 post-diagnosis. And also we haven't looked very closely at that group. So we don't know, I will say, is the answer, because the goal really was to look at 19 negative relapse, and we didn't have a lot of patients. And then, as you know, there's the whole, I'm only focusing on the leukemia part of it. There's the whole other part of like the T cells. And because this is a T cell therapy, so I think the thought is that for patients that have a CD19 positive relapse, it's more a failure of the T cells, either to persist or to really get, somebody into remission. And so that's a whole nother part of the story that we don't have data on.
Questions? Okay.
I can start maybe with a short question.
Yeah.
Not so short, because at this point opens the problem of looking at the microenvironment, which
Totally.
is do you have any idea on how to apply this system?
Yeah, to the microenvironment.
To the microenvironment.
Yeah, so I think, once you can certainly do this kind of imaging in bone marrow. The harder thing, I think, is getting bone marrow biopsies, right, from patients, because that's not typically what we save. But that is one potential route, is to try to understand, how can you look at this in the context of the microenvironment, in the context of where the cells are in the bone marrow and what's going on there. Yeah. So the question is, can you look at the peripheral blood the same as the bone marrow and say that you're basically getting the same cells? So we haven't been able to look at that in a really statistically robust way, because, as you know, oftentimes for patient samples, unless you set out to ask a question and collect the samples along the way, otherwise you sort of get what you get. We did in the Nature Medicine paper, and it's kind of buried in the supplement. For some of those patients, we had peripheral blood and bone marrow from the diagnosis. And they're not exactly the same. There are some markers, and I can remember, I think, like CD24, and there are some markers that are different between the cells that are in the peripheral blood and the cells that are in the bone marrow. We don't know the presence really of like the relapse predictive cells if compared blood to bone marrow. But that is a really good question. And I tend to think that, like, cells going into the peripheral blood versus staying in the bone marrow is more of a reflection of their biology. Like, are they tuned to stay in the marrow through integrins and other cell-cell contact things versus are they permissive to go circulate in the blood? So that's a great question. I mean, it's interesting. There is a clinical trial, which I know Monza is participating in, that we are as well, using CAR T cells early, like upfront. We'll see how that looks, but I will say that the data, as far as I know so far, looks pretty promising. The question I think is going to be, because I do think what's going to happen is these targeted therapies are going to move earlier, and they already are. Like, you know, blinatumomab, inotuzumab, all these targeted therapies are moving up front. I don't know where CAR will go because it's expensive. If we end up getting like an auto or an allo CAR off the shelf that's actually worth it, you know, that's possible. The other thing that's interesting is actually, I think it was a European group that published an adult pH positive ALL, you know, basically getting rid of a lot of the cytotoxic chemotherapy and just doing steroids, dasatinib, and then blinatumomab, right? And having like very impressive response rates. Now, whether or not there's going to be durable response with that approach, we have to see. But I do think that there is the opportunity to I won't say de-intensify, but change. Because the therapy that we've been giving to kids with ALL for 50 years is about the same. And it's 3 years long, 2 years, 3 years long. And it's very, you know, it's hard. And the kids don't go unscathed. They have long-term side effects. So, you know, our hope, I think, is that, you know, combining knowledge of like, what are the most important cells to treat and what's the best way to treat them, that then we can make those decisions early and offer less toxic therapies. And then also patients who don't need them, like the kids who don't look like they're going to relapse, de-intensify their therapy. But doing that actually in clinical trial space will take a long time. Yeah. So the question was, you know, how do you manage the different expertise required to do this type of data? And it's a really good question because it's definitely challenging. Because I'll say now in my group, and I learned this from Gary, that 60% of everybody, 60% of people are sort of wet lab biologists, and 40% are computational. You know, and I have basically 2 people who are almost purely computational. And then everybody else sort of either has a little bit of those computational skills or a lot, actually, you know, and or you collaborate. And even for me, I'm a clinician, you know, I'm a physician and not a trained computational biologist and so I've had to learn how to find people who know what they're doing and go ask them for help. But it has been fun to get to, like, I can't execute any of it. Like, I can't code, I can't do any of it, but I can know what are the questions I want to ask, and I can know a little bit about how should we approach them. And then you have to sort of have this conversation about your different expertise, because the purely computational people will love to like make a new method, or, you know, oh, I found all these things that are important. But then when you come to the biologist or the medicine side and you say, well, that's great, but I can only handle 3 things. So like, what's the most important 3 things? Or this doesn't mean anything in biology. So it's a really intricate dance, and also it's fun because you learn from each other. But I think in science now it's very hard. It's hard to be an expert on everything. So you have to, you know, like, I don't know anything about splicing. I don't know anything about metabolism. So you start learning, right? Like, and then you go find people who know. Yeah.
Thanks a lot, Cara. With this
Thank you.
intricate dance, we can stop and have a lunch together. Thanks a lot for everybody coming, and thank you.